So we continue with the embryology of the nervous system. So actually, the human nervous system is a marvel of complexity, controlling everything from basic reflexes to conscious thoughts. But how does it all begin? So we'll basically look at the timeline, and this is an overview of how the nervous system is being developed. So we explore the embryology of the nervous system from its origin to the intricate structures that it forms, the brain, spinal cord, the nerves, virtually everything. So here, remember that the development of the nervous system begins in the third week of the embryonic life um, with a process called neurulation. Okay, so with the neural plate formation, the ectoderm actually thickens along the middle line to form the neural plates. And the first indication, that's the first indication of the nervous system. And remember the neural groove we talked about, and the neural folds. So the neural plate invaginates to form the neural groove, as flanked by the neural folds. And by the end of the fourth week, the neural folds fuse to form the neural tube. As the cranial end develops into the brain, the caudal end develops into um, the spinal cord. Because the brain develops from the cranial portion of the neural tube. By week 5, three primary brain vesicles appear. The prosencephalon, that's the forebrain, gives rise to the cerebral hemispheres, the thalamus and the hypothalamus, and the mesencephalon, that's the midbrain, which forms structures involved in vision, hearing, and motor control, and also mesencephalon, that's the brain, which also develops into the cerebellum, the pons, and the medulla. Okay, so these vesicles further divide and shape the brain's intricate regions. So the spinal cord, the caudal portion of the neural tube becomes the spinal cord, and cells within the neural tube differentiate into various key structures, like the neuroepithelium, which generates neurons and glial cells, a mantle layer, which forms the gray matter of the spinal cord, marginal layer, which becomes the white matter, carrying signals up and down the cord. And with the neural crest cells, um, whilst the neural tube forms the central nervous system, the crest cells actually give rise to many essential structures like the peripheral nerves, the schwann cells, the adrenal medulla, a part of the craniofacial skeleton. So they migrate throughout the body, demonstrating their dynamic nature of development. But the key processes in the formation of nervous system, think of the cell proliferation, with the massive growth of neural and progenitor cells, cell migration. The neurons move actually from their final locations, and they move to their final locations, axon guidance, and also synaptogenesis. So with the axons, they navigate to form precise neuronal connections, and when it comes to synaptogenesis, the synapses form, enabling communication between neurons too. Alright, so as you can see over here, third week, we have your blastocyst embryo, first month, as a fourth week to ninth month, to second month, and then with from the third month to the ninth month, we are your fetus. But fourth week, as you can see over here, the features and swellings distinguish the forebrain, that's the prosencephalon, from the midbrain and the hindbrain, the midbrain mesencephalon, and the hindbrain organcephalon. And the fifth week, you see over here that the nerves grow into the body wall, autonomic nerves grow into the viscera within the body cavities. By the end of the fifth week, like I said, the forebrain divides into the um, diencephalon and the telencephalon as a future cerebral cortex. And also the third month, You have a cerebral cortex, the telencephalon overgrows the midbrain, and by the sixth month, you have a primary gyri beginning to appear, and your secondary gyri present. The brain is like 25% of that of the adult size, but always remember that there are other defects, clinical correlations that you can think of, as if there are disruptions in the development. So it includes neural tube defects like spina bifida, anencephaly, which results from improper neural tube closure, microcephaly, which is caused by reduced brain growth, um, holoprosencephaly, as a forebrain defect leading to incomplete division of hemispheres, and also hydrocephalus, that's excessive cerebrospinal fluid in the brain due to impaired drainage. So the nervous system is actually a masterpiece of embryonic engineering, with every step intricately um, choreographed. So understanding this development helps us to actually appreciate the beauty and complex of life. So yeah, so basically that's just an overview of the nervous system. Embryology. Okay, so with this, okay, you can see the oropharyngeal membrane, orbicopharyngeal membrane. That's the appearance of the neural plate. The notochord, of course, over here, paraxial column, paraxial mesoderm, intermediate mesoderm, and the lateral plate mesoderms, the somatic endosplans, nucleus, and over here you can see the placos, the lens placos, olfactory placode, hypophysis placode, the optic placode, forebrain, midbrain, hindbrain. Have your axial rudiment, spinal cord over here, neural crystals forming this layer. So as a developmental phase of the local region of the ectoderm of embryonic discs at 18 days. But here you can see the neural crystals and the notochord over here. Of course, somites, for example, mesoderm, intermediate, and then the lateral plates, mesoderms, and there's the intra-embryonic cellum. And okay, the neural tube, the neural plate forming the neural tube, neural crystals. Okay, so for neurulation, as you can see over here, neural plate of the forebrain, the neural groove, and the folds, neural folds. See the occipital formites over here too. Okay, if you make a section over here, you can see the ectoderm, the neural crest cells, the future neural crest cells, and neural plates. It's the same for this section over here. You see the neural groove that is here, same over here, and then the neural folds, which will become the neural crystals, and then yeah, okay, there's a primitive streak over here. Neural plates of the forebrain, neural groove. If you see the fused neural folds, the neural folds over here, the groove, FS, so I call formite. And we take a section over here, these are the neural crystals, and the caudal neural pore is also over here. As the embryo at 21 days, like 3 weeks. Okay. So the neural folds are fused over here. The first occipital formites, first cervical formites could be found over here. The thoracic too. And there's a caudal neural pore. And when you take a cross section of this, we see the ectoderm, the neural crystals, and then the neural tube being this. See the sulcus limitans too over here. Okay, so ectoderm, neural crystals, neural tube, that notochord, and As this is the embryo 24 days. By 6th week, you could see all these. So let's take the crystals forming the sensory neurons of the dorsal spinal ganglion to, you see the mesonephros, then from the intermediate mesoderm. You see the sympathetic trunk ganglion, the spinal cord, dorsal spinal ganglion, the pre-aortic sympathetic ganglion. Okay, that's over here. That is the aorta, and that's the cortical primordium, and you see the dorsal mesentery. Of course, that's the gut tube, and germinal epithelium of future gonads too. You can see them over here, and yeah, this is the serosal lining, peritoneum of the abdominal cilium. This peritoneal cavity. Okay, this is visceral motor neuron of the sympathetic ganglion. It's chromaffin cell or suprarenal medulla cells too. The neural tube defects, you could see spina bifida occulta too by here. Sinus with squamous plaque over here too. That's the dural sac called the equina. So basically, it's a fatty part overlying the spina bifida occulta, and tuft of hair or always keep them for may be present, or maybe there may be no external manifestation. So dermal sinus also present in some cases. That's the dermal sinus you could see. There are several types of spina bifida. So you could see the one with protrusion of the contents as meningocele. You can see it over here. The meningomyelocele. See part of the chorda equina, spinal cord being in the meningocele. Okay, neural tube defects, the occipital encephalocele. Okay, there's the one over here too. You can see, and there's a frontal encephalocele, anencephaly too. Okay. So neuron development, two postganglionic autonomic neurons of the sympathetic or parasympathetic ganglion. We see over here the satellite cells, the endings of the preganglionic autonomic neurons now absent with the cell bodies of the postganglionic neurons. See the myelin sheet, neurolemma. It surrounds the unmyelinated axons. Okay, these are two neurons that we are talking about over here. Okay, the axon endings on the glands or smooth muscles. So on the glands over here, smooth muscles, cardiac muscles over here too. So during development, as you can see over here, the dividing satellite cell, that's the neuron cell body, and Schwann cell is a neuronal cell over here too. The neuron endings of central process with spinal cord or the brainstem, as the neuron endings with the peripheral process within an organ. Okay, so that's during developmental stages too. But when it becomes matured, if you see the nodes, you see the myelin sheet, you see the satellite cells surrounding the soma, the neuronal cell bodies, and nodes within it. Okay. Okay, and that's the development of the cellular sheet of the axons. See the axon over here. Okay, of course there's the axon, but there's the Schwann cell, the peri-axonal space in between. That's for the unmyelinated axons of the peripheral nervous um, systems neurons, and then for the myelinated, you see that covering, it moves all the way around it. Okay, and that's similar to that of the central nervous system too. Okay, and that's the oligodendrocytes. Okay, so remember the development of the spinal cord layers, like we talked about. Ectoderm, neural crystals, neural tube, sulcus limitans. Then with the development of the neural tube, we have the central canal. That is, there's the migrating neuroblast and the pia mater. So that's ependymal layer of the mantle zone and the marginal zone. Mantle zone being the gray matter, and then the marginal zone being the white matter. Marginal mantle neuroepithelium. Then you have the canal with the internal limiting membrane. You have your neuroblast cells all through the neuroepithelium and the mantle layer. And external limiting membrane, that's where you have your PL cells, you have your pia mater also. Okay. Is it a central canal? So the 5.5 weeks, there you have your central canal over here. Then the ependymal layer, the mantle layer, and the marginal layer. Sulcus limitans to over here too. Okay, that's essentially the coordinating part of the dorsal pleats rather, and then that's the ventral pleats or the ventral basal pleats, usually motor, the dorsal being sensory. So with the dorsal, okay, this is a more mature transverse section. Is it the central canal, the dorsal gray column or the gray horn, the white matter or the tracts. That's the lateral gray column, the ventral gray column, the tracts, still white matter. The neural crystals, as you can see, it's right over on top of this, the tube. The peripheral nervous system too. The differentiation group at 28. These, you see the spinal cord, thoracic part of it. Sensory cells from the neural crystals and commissural neurons, association neurons to over here, and the ventral funiculus also over here. So there's a detailed diagram too of the peripheral nervous system. So the growth of the neurons from the 5th to 7th weeks. You see all the various parts of it, the central, the dorsal roots, and then there's a preganglionic, synaptic, and motor neurons. You see the funiculus, the lateral, the dorsal. Okay, you can see this is definitely the ventral, and the ventral roots ganglion, dorsal root ganglion, those all is it dorsal root ganglion, but ventral roots, there's no ganglion over here, remember. Okay, this is a sympathetic trunk and the ventral ramus of the spinal nerve. Lateral sympathetic triganglion. Right. So all these happened acquired by 28th day only, and with the peripheral nervous system, that's quite simple. You see the gut tube, you see the neural crystals, the neural tube, the migration of the crystals too. Okay. The dorsal roots, ventral roots, dorsal root ganglion over here. You have your epixyal muscles, the dorsal ramus, the ventral ramus too. Epixyal muscles, the flexor muscles of the limb there. Okay. So with the growth of the spinal cord in the vertebral column, you see the diagrams clearly over here too. And the embryonic dermatomes, occipital, cervical, thoracic lumbar dermatomes too. They're all clear over here, as you can see. With the adult dermatomes, C2, C3, C4, C5, from T1 all the way down to T12, to C6, C5, C7, C8, L1, L2, L3, L4. Posterior part you can see S1, S2, S3, S4, S5, and the L5, L3 over here, L2, and L1 over here. So with the development of the brain, like I said, the forebrain, midbrain, hindbrain, spinal cord over here, and the various divisions, as you can see here.